Okay, thank you guys. I'm really honored to be here. I'm also a product of UGA and MCG, and I'm going to talk to you guys today a little bit about lasers. Just get this set up a little bit. Okay, before I begin, I just wanted to give you a little bit of orientation of where I came from and where I'm at now. I graduated in 1993. I had the distinct honor of being the last person in Lloyd Logan's scientific illustration class and the first one in Gene Wright's. And remind me to tell you a story about that later on. I went to the Medical College of Georgia, graduated there in '96, and really had another great opportunity to be one of Kip's interns at the Educational Resources Center in 1996. And a lot of what we're doing down in Grenada, as I was telling some of my colleagues, is modeled after the ERC. I think it was very inspirational to me when I started there, and I'm really excited to see how sustainable it's been over the years, and I hope that happens as well down in Grenada. And if I don't want to forget, I have a few more thanks before I go. Without Jessica Holland, I would not have even been up here to give this talk, and she was my first director down in Grenada, and thanks for taking a chance on me. Okay, so where are we at in the world? Where is Grenada? Grenada is in the southern Caribbean. It is a small island nation in the West Indies. And when I say small, it's pretty tiny. It's a three-island nation, although there's a lot of different little islands in between, some uninhabited. But the inhabited islands are Grenada itself and Carriacou and Petite Martinique. And there's a few active volcanoes, I hear, underwater near there. And Grenada itself, just so you get an idea of how big it is or how small it is, it's about, 12 miles wide by 22 miles long. And if that is, basically, if you know anything about, let's say, like Lake Tahoe, it could fit comfortably in Lake Tahoe, basically. So it's like living in a small town that you cannot escape from, basically. Not easily. And it has a lot of charm. It has a lot of pros. It's been a very profound experience for me. And any of the struggles and the hardships have definitely paled in comparison to the opportunities and hopefully a lifetime of memories. So the talk today is inspired by the jungles of Grenada. So Grenada, like I said, is very formed by volcanoes. They have a crater lake in the middle of a rainforest national park called Grand Etang. And this is me hiking up to a vista called Mount Quaqua. And the jungles themselves are actually pretty benign. I would say that in Georgia, it's a lot easier to get stung or bitten or killed by something than it is in Grenada. I mean, and I'm not kidding. It's like there are some monkeys, and supposedly there's snakes, but there's nothing really poisonous. And so it's incredibly rich and diverse, but not too scary to be hiking around in the jungle there. It's got a lot of geothermal activity. So that orange stuff it grows, the algae grows because that waterfall itself, there's like a little pipe that's coming out of it, and it's like this warm, fizzy water that comes out of the ground. And when you're hiking up these rivers, you can see the water bubbling from carbon dioxide, I think. So it's a really lush and interesting environment. This is where I work. This is St. George's University. This is a new view of the campus. The campus has gone through a lot of growth over the last few years. There's a new gym and a new facility there. And in this shot, we actually own, because this is a drone. Here I am. My colleague Brandon Holt was very nervous flying his drone over the ocean that day, and we got the shot, though. It's great. So let me just talk to you a little bit, or let me tell you a little bit about the art. So the campus itself, there's about 2,000 faculty. There's 3 schools. There's a School of Medicine, of course. We work in the Department of Anatomical Sciences. And there's a vet school that we are very, very honored to be collaborating with any chance we can get, and a School of Arts and Sciences that we hope to convince that the word arts in the arts and sciences is really important. And so we're working with about 7,000 or more, actually 7,500 or more students on campus. So this is my team. This is the current version of our team. And several of us are also products of MCG or UGA or both. So that's me, David Nahabedian, Brandon Holt, and one of our newest faculty, Joshua Hatfield. So all from I'm proud to say, all from one of the Georgia programs or both. Leaving 3 more Americans on the team. Sue Simon, who graduated from the BBiz in Chicago, and also Valerie and Jack, who are also American, but they are graduates of the undergraduate medical illustration program at RIT. And that leaves our European and other country counterparts. So Claudia Carceles comes from Spain. Claudia and Sarah, I found they were graduating from a program in Maastricht in the Netherlands, and it is a Master's of Science illustration. And I also found that there is an undergrad, a bachelor's in natural history illustration in Newcastle in Australia. So Lucia Garces grew up in Colombia, but she went to school as a graphic designer in Colombia, and then moved to Australia and practiced graphic design for about 12 years, and decided she loved natural history and science illustration. And so she graduated from there and applied for the position, and we're very, very happy to have a professional graphic designer on our team. And in fact, she designed this PowerPoint presentation, so I should give her a shout out. And then Faria Khan, I was able to start an internship program. So several of the School of Arts and Sciences programs, especially biology, have to complete 160 hours or something like that for their, as part of their for graduation. And so Faria actually came up to me and she goes, "I want to do what you do." And she's from Trinidad and Tobago, and so she agreed to be the guinea pig for our first intern. She completed 160 hours, and then she just wouldn't leave. And she stayed on for the entire year last year, and she came to me and asked, how could she possibly, maybe even get a job there. And I said, well, just become indispensable to us, and that's what she did. So we were very proud to hire her last term as a junior member of our team, which is something that is our goal, is to hire more and more local Caribbean colleagues. So here are some of the projects that we do. We work on research illustration. You guys know what that is: conference posters, journal publications. We have several PhD candidates who need their illustration work. They need their research illustrated, I should say. So that was really the bread and butter. When Jess first hired me, that was a lot of what we did. we worked with the Anatomical Sciences Research Fellows, which are MDs who had just recently graduated. They haven't quite got their residency yet, and they're working in the office with us, writing review papers for various journals, trying to give a good name to St. George's University and also get them an edge when they go and interview for residency. And of course, to get published, an edge in publishing, having some professional illustration is always a good idea. And so our dean, Dr. Marios Lucas, really devised this whole idea of hiring Jess and starting the medical illustration program of partnering up researchers with illustrators. And it was, of course, a brilliant idea that has really evolved. So the main meat of what we were doing was research illustration, and then when I got hired, Jess and Dr. Lucas were talking about doing larger projects, something that Jess couldn't handle necessarily as an individual, and they wanted to do a lot more book projects. And so about 4 years ago is when I started, and some plans for both of these books were underway. There was a first edition of the Gray's Clinical Photographic Dissector, and then Jess and some other of our colleagues had already designed a prototype for the ultrasound anatomy book, and so me and 4 others, we worked really hard over 2 years or so to create these 2 books, and I'm really proud to say they were published and came out last year. I'm starting to see this clinical photographic dissector used by students around the campus now. Then Dr. Lucas came and said, how do I hire 2 or 3 more medical illustrators? Some of you guys have seen that we constantly are having openings up on the AMI Post. And one of the reasons is that we're growing, and the university is growing, and so Dr. Lucas is our patron saint of medical illustration. He wants to also grow our capabilities. We were able to convince him that instead of bringing in 1 or 2 or 3 medical illustrators that have already got their graduate degree, is that maybe we could open up a fellowship for those who graduate, say, from Gene's class or some of the other scientific illustration programs around North America and the world. And luckily, he went for that, and so now we have been able to hire those teammates that I showed you. Some with master's degrees, some with bachelor's degrees. I mean, we all work together as one unit, but we're also split into sort of the research division and then this fellowship division. But the lines are really blurred. We're often working on each other's projects. So the fellowship was conceived to do exactly what my colleagues before were talking about, the fact that lecture all over the world, I think, or at least in the Western world, lecture hours are being cut at universities and replaced with online learning, what's called directed learning activities. And Dr. Lucas saw this as an opportunity to start creating these kinds of things in-house. And that's what we've been doing. So the fellowship, the fellows in the fellowship, their main mission is to create the directed learning activities. They create 2D animations, 3D animations, just reorganizing, cleaning, and illustrating PowerPoints for faculty. I tell them that we are in a position where we can take risks and we can really try to engage students in a lot with humor and a lot of fun. And so they really get a chance to, express themselves in a lot of ways and kind of branch out from your traditional illustration projects. And speaking of, we have a vet school. It's also a very, very, we're honored to work and collaborate with the vet school, like I said, whenever we can. Jessica was doing this early on, and we've continued the tradition of creating, these body paintings of anatomy on equine and bovine. We have not attempted this on a dog yet. But we will do this. We'll wake up at like 6 in the morning, and we'll be over at the Large Animal Research Facility. We bust out these illustrations on the sides of horses and cows, and then faculty will come in and do lectures, generally about palpating and auscultating. This term, we're going to do one with let's see, basically with ultrasound, and it's been really, really fun. And so what I hope to see is that we're going to evolve and do a lot more with the vet school as we move on. But really the talk is about lasers. And we had a chance, my other colleague, Sochi Vanaha, she practices facial prosthetics, and I don't know, if you work at a university, sometimes you get a chance to put in a wish list, and she knew another colleague who was using this Artec Space Spider, and so we just put it on the list. We didn't think we were going to get it, and it showed up, and that was not, then that's where this talk kind of starts. It's like, what were we going to do now? So the Artec Space Spider, if you've never heard of it or you never use it, so it looks like a blue iron, like almost literally. It was supposedly originally designed for the International Space Station. It's supposed to work in a lot of different climates, which seemed attractive to us. Basically, though, when we bought it, it was around $28,000 US. So again, we thought we would not get this thing. But it showed up. And then we realized it also needed a few other things, like a gaming computer to run it. So that took a while. It sat on our shelf for a little while until we ended up getting that going. Full disclosure, it's not really, so this whole talk is a sham. It was not really lasers, but structured light, blue LED light. There's a lot of pros for using that type of technology. Structured light is based on triangulation, so this scanner has a whole bunch of cameras, and it's shooting out this pattern of light onto the object, and it has a pattern that it knows, and then whatever distortions that happen, it detects the distortions as it falls across the object by the surface details, and then that's what it reads in. So it can shoot many points of light super fast, and it's incredibly precise. So, like I said, for the longest time, we were waiting for the other parts of our setup, but this is the basic setup, and it's really easy to use. Probably the main thing that we had to wait for was the Alienware to show up, and we had to convince someone that we were not playing video games on it, but, well, I want to talk about that. But we definitely find this thing incredibly easy to use. And we set up just a $5, $10 Lazy Susan. My colleague David Nahbedian is pretty handy, and he created that stand. We started out doing a lot of stuff with plastinated anatomy and tested out with that. It's super easy to use. It's really efficient and really fast. And I'll just show you a video of us doing a scan here. I mean, essentially, you point it at the object like a video camera and start spinning it around. You could also just kind of wave it around as well. You're not limited. We had a trainer come down and show us how it was done, and we were so careful and cautious with it, and he was just like all over the place. And it's super fast, super easy. And what we found is what was taking, I don't know, like, we were using a lot of photogrammetry before this, and it would take, 8 hours to do a photogrammetry shoot. We would send that either with our, at the time, we weren't using Agisoft PhotoScan. We were using whatever descendant of 123D Catch of the day was. We were sending that off, and we'd wait for 48 hours, and then we'd get a model, and then maybe we'd had something that worked, or maybe we didn't. So now it takes us about an hour to create a very accurate and very precise model, and with photographic textures as well. And it's supposedly accurate to the on the micron level. So if you look at the numbers up here, it doesn't necessarily show that. I think a lot of shrinkage, and I'm not sure that we were measuring exactly, but it's incredibly precise for what we use it for. And it's safe to use. I mean, we've done a lot of scanning of each other in the office. We generally don't keep our eyes open just in case. I'm a little afraid but David was pretty brave, and we scanned him, and we noticed it caught even just the hint of edge of his iris, but of course it didn't pick up the cornea. So we've been playing around with creating 3D portraits in the office. My colleague Claudia took that assignment and really ran with it. So we did a whole photogrammetry of each other just to train them up on photogrammetry techniques. And then they did a mashup. Each person also had another colleague scan their face, and so she put those together, and also was training in Horos and created a model of her own brain. So I thought that was a pretty amazing application for the Artec, along with the other 3D capture technology that we have in the office. We've been playing around with just scanning anything and everything that we can find. I think over lunch we were joking with some of my colleagues, some of my newer colleagues, about how it's not too weird for us to have like bugs and dead bones, dead animals and bones and stuff in their freezer. My medical colleagues don't understand, but one of the freezers they open up, it's pretty scary. It's getting worse every day. But we've been playing around with scanning anything we can find. Katie Yost also was a member of the ERC, the certificate program, and we hired her from the ERC. She'd go out running and she'd find these goat skulls and anything she could, and we would practice scanning with it. So I can tell you a little bit of its limitations as well. First of all, of course, it only captures surface details, so that's fine. As opposed to like CT scans. On the flip side, it does have an onboard camera, so you get photographic textures as well. But there is a lot of cleanup. David especially wanted me to tell you guys that most of the things that he scans, he does have to spend several hours cleaning up. So even though you get a lot of great detail, there's always an overhang or there's a crack or a crevice that you just can't get into with this thing. And so having some ZBrush skills, having some modeling skills, of course, is key. The scanner itself, scanners in general are confused by wet, shiny, clear, or reflective surfaces. And so that generally is a problem for us when we're scanning in the lab. Pliable, sagging, loose materials that change shape are a problem because it cannot find the reference point once it moves. Long bones like the femur, anything that's flat and or the same shape for a long time with no recognizable features, like the femur, or anything that's really flat or thin, like scapulas, it really can get very confused. And very thin objects like wings, hair. It had a claim that it could get down to 0.1 microns as far as detail. And I don't know if we're not exactly doing it right, but we haven't been able to get that much detail yet. Our classic test was with this piece of chicken that we bought on campus. That, of course, gave us a lot of, it really was a minimal viable product for us scanning in the lab with the cadavers and the prosected dissections. What we settled on was hanging it from a line and letting gravity work with us instead of against us. And so that actually worked out really well. But we have noticed that we're seeing some really weird artifacts with scanning faces and skin. We're not exactly sure if that is just the properties of skin, the translucency of skin. Most likely it's a combination of stuff, and probably oily skin is reflecting. But so this is some of the cleanup we have to do, is to watch out for this stuff and kind of clean it up. This is one of our newest colleagues who I think just recently graduated from Gene's Science Illustration program, Josh Hatfield, and then our newest intern, Carol, she's from Syria. Her fingernails were too shiny. You can see how they kind of got a little dented. And we joked that we'd have to use sandpaper to sand down her fingernails. She wasn't really into that. But we do. We use a lot of sandpaper, and we also use this German 3D scanning spray. Work really well. Basically, you can use even athlete's foot powder, so anything that like sprays this sort of matte thing. So as a metal badge, there's no way that would get scanned. It was hard enough by how thin it was, but spraying it with this stuff, of course, sets it up for success. And here's just an example. I mean, there's just no way you're going to get all the detail, even though the scanner does an amazing job in general. Getting into trying to get these conchae is just not going to be possible. So if that is what you're after, you're going to have to do a lot of rebuilding and sculpting on your own, but it gets you an enormous start on your project. And this was our first test that we knew that we're not going to be able to get so much detail. We tried scanning this grasshopper, and we just couldn't get it. It really, that was the best we could do with that. And just, it has a range, and that range is just a little bit too small, especially these little thin structures. So it does have some limitations. I just recently have been trying to repair my sunglasses that I broke, and I'm happy with the results so far. I sprayed down the actual unbroken arm, and I got pretty close, but you can see, again, some of the limitations. It's not going to get into those holes, and it's not going to, you're going to have to do a lot of cleanup. So the other part of the Artec scanner is the software itself, the Artec Studio. And it's got a lot of powerful features and algorithms that have really updated from its version that we got at the beginning, which is 10. So the Artec Studio 13 has made some improvements. They got better hole filling, making better watertight models for 3D printing, a simplified and more organized mesh. It's kind of hard to see here, but we had scanned an open heart dissection, and we scanned it a while ago, and we ran it just this week with the new software, and it seems to have even better. So it's amazing that the scans we did a while ago, when the software gets better, the result is even better. It also has an automatic feature. So before we had to go through each click and we had to set up everything, we had to go through all the process, and now it has an autopilot system by which you just press a button. You have all the things set up to your preferences, and it will make a model for you. And that's something that has been really beneficial to us. So what are we doing with it? We are doing a lot of illustration work. We are a firm believer, do not stop drawing, for God's sakes. Don't stop drawing. Keep up your traditional skills. But if we have a chance to create a 3D model, we're going to do that because of the versatility of it. And so if we have the time on a project, we're going to create, we're going to take the time to scan and create a 3D model, and then we'll turn that into an illustration. This is one of David's for an upcoming strokes project. Katie as well. David scanned this plastinated cerebellum, and then we use that in the illustration work as well. We're also partnering up with the vet school. We've got 3 3D printers. I brought down a Taz Mini, which was a very, very interesting process getting that through customs. They did not know exactly what I was trying to bring into the country. And that inspired our dean to purchase a couple more 3D printers. We also have a Formlabs Form 2. And mainly what we're doing, of course, one of the main things we do, it's really basic. A lot of you guys are doing this. is we're just scanning a lot of the more uncommon species and more uncommon specimens, and we're creating some life-size replicas that students can study, and also they won't break them, or if they do, we'll print them another one. Yeah, so from dissection to interaction, from lab to SKETCHFAB, we're doing a lot of stuff with this work. We have some, if you haven't heard of SKETCHFAB, we've got and I know I'm about to go over time here, but we have been deploying these 3D scans in the classroom and also for specialty publication work. Dr. Marcos came down and did a full dissection of the bovine heart conduction system. He wanted just an illustration, and we showed him what we could do with 3D scanning, illustration, and also bringing it up into SKETCHFAB. So moving forward, we are very interested in taking what we've learned over the last few years into creating what I'm now calling mid-fi simulation. We're highly involved in the upcoming INVEST conference that's going to be hosted at St. George's University in May. So INVEST, if you've never heard of it, is the Innovation in Veterinary Education Simulation Technologies. And we're working with several of our colleagues, our vets that are writing research on up in their game in lo-fi simulation. We are just finishing up scanning a pit bull skeleton from tip to tail, from the ruta to the tuta, as they say, and we're going to be putting that into some here, I'll just show you. So we're basically going to be creating some joints and especially hip problem simulations for the students. So if you get a chance to come down to Grenada in the May 1st to the 3rd, I'm really proud to announce that we were able to score David Kilpack, who's one of our close colleagues in the AMI. He's going to be one of the keynote speakers, and he's going to come down. And he also, we've got him scheduled up. We're going to do some turtle research. There's a leatherback conservation group that's at the north end of the island. And you go and you patrol the beaches all night long, and you help them lay eggs and protect their nests. And it's a pretty amazing experience. So I'm very, very proud to have David along with us in May. All right, thank you guys for everything. I'm really excited that this is happening, and it's an honor for me to interact with you guys, my newer colleagues, my young at heart colleagues that I've known through the years. Thank you guys for everything. That was Lasers in the Jungle.